Welcome everybody. I hope you had a nice free day yesterday. Um, okay. Amelia, are you here? Because I think you're missing. Okay. Anyway, we'll start. I'll wait 1 second more. Okay, let me tell what we're going to do today. First and all, I want to make a little announcement for tomorrow. I have some news that I think is interesting to share, like world news, let's say. And then we will have a look into the cell organelles. It will be a very superficial look into what the cells are made of, because the objective of today is basically that you recognize the parts and that you know their function. We don't have to know details today. That's really not that important. We just want to get familiar with the most important parts of the cell. And like you can see in my background today, it will be mainly about the plant cell and the animal cell. Okay, and then we will finish with a little activity, very simple activity to see if you understood everything, and I will share that with you. So the first announcement is that what I already put in the announcement, and I put it in our WhatsApp group, is that tomorrow uh, we will not have a class at 1 o'clock. We will move the class to 11. So you don't have to connect at 1 o'clock. I will take absences at 11 o'clock. So the class, both classes at 9 and 11 will be substituted by our guest speaker, who is only able to do her call, her conference, let's say, 1 time. So that's why we joined the groups, okay? Because unfortunately she has not the time to do it twice. And then that's it. So that's the announcement. So tomorrow we see at 11. So make sure you are there. And then at 1 o'clock, no lessons. Okay. And today I would like to start the news with Amelia. Right? Perfect. The news I wanted to share with you is the following. 2 days ago, all the newspapers were full with the news that they found possible signs of life on Venus. Okay. Venus is our nearest planet, the closest to Earth. It would only take about 108 days to get there. Mars is way further. But in fact, Venus has never been a candidate for life in our solar system. It has never been considered a candidate because the surface of Venus is so hot that lead would melt. The metal lead would just melt on its surface because it's very, very hot. It's more than 300 degrees. Look, you can see here 470 degrees, you know. It's very hot depending where you are exactly. And another thing is that the atmosphere is 96% carbon dioxide. So it's like a global warming effect that is like a million times stronger than on our Earth, because on planet Earth we don't even have 1% of CO2 in the atmosphere. We express it in parts per million, and we are around 400 parts per million, and we used to be around 300 a century ago. And then the other thing why scientists never thought that life would be possible is that the pressure is really, really high. So the pressure is 90 times greater than on planet Earth, so it would mean that you're almost a kilometer in the ocean at that depth. So it's really high pressure. But it's not on the surface of Venus that they think to have found traces of life. It's in the atmosphere. So the atmosphere that is the closest to the ground, it's very high concentration of CO2. But the higher you go, this atmosphere changes in acid. It's basically sulfuric acid. It rains sulfuric acid. There is other gases there. And at a certain height, they found above the acidic level, they found a molecule which is called phosphine. Okay, and phosphine is not life. It's a biomolecule. Where did I see it? And phosphine is interesting because it's been associated with life. So they think it's interesting because here it is, because this phosphorus atom with the 3 hydrogens around has only been associated with life. So wherever there is phosphorus, it should be like some kind of byproduct from bacteria. So now it is possible that it indeed is a byproduct of bacteria, and that there might be some microbiological growth in the atmosphere of Venus. Okay, it's definitely not intelligent life, but it might be that at a certain level on Venus there is in the atmosphere this microbiological life. Scientists, they believe that once, a very long time ago in history, there must have been liquid water on Venus. And another thing that is very interesting is that the composition of Venus is very similar as the composition of Earth. I mean, if we talk about atomic elements, you know, I mean, of course Venus is Earth's evil twin sister, because it's such a harsh place to life to harvest. But if we talk chemically, it's not that different as planet Earth. It's just way hotter, and it's different distributed. So it is possible that this life that might exist on Venus, that it's there for millions and millions of years already, and that it possibly started billions of years ago, and even on the surface, but now on the surface is not possible anymore, and it's only in the atmosphere. You cannot compare the atmosphere of Venus with our atmosphere. It's so dense, it's so thick, that you have to see it a little bit as something between water and air. You cannot see it as air. So it's possible that it's so thick, this atmosphere, that those microbes are floating in the atmosphere. This is Why is this important? Basically, it's very important because if it would be true that they find microbiological life in the atmosphere of Venus, then extraterrestrial life is way closer than we ever thought it would be. And if there is life on Venus or any place in the solar system, then it would mean that life is at most likely very abundant in the universe. And this is something that I really think that life is abundant, but we just don't know how it goes from one place to another, and maybe there's some burden to it. So let's see. I think that in our lifetimes we will get some news about this. We will be told the confirmation that there is life out there somewhere, some kind of life. Or maybe we can find proof that life has been on another planet, that we find proof that life got extinct on another planet. I think this is a good possibility, but we'll see what happens. Okay, but I wanted to share this with you because I think it's really cool, especially if you think about the origin of life. um, think about the Miller-Uller experiment. I mean, you just take the right atoms and you put some lightning and some heat and some electricity and whatever you want, and you already see biomolecules appearing after certain hours. So conditions on planet Earth and other places in the universe, they have been subject to similar conditions, which makes it very likely that some kind of life might have developed in other places as well. That's truly what I think as well. So somebody wants to tell something about this? Otherwise, we can move to the essence of the day.
Me, um, will it be possible to recreate this environment like artificially with electricity and some chemicals to
Uh,
create new forms of life?
yeah, I think there must be possible. In fact, it would be a good experiment to do. But the thing is that if you have biomolecules, that doesn't mean yet that you have bacterial life. Moreover, to go from biomolecules to bacterial life or single cell organism, it took billions of years, or even millions or billions of years. So it's not an experiment that you could easily set up in a laboratory and just put it all together and see if life appears, because it would take more than a human lifetime to do that. So it's not the best way to check this, but I'm sure that somebody set up a certain experiment like that and they're waiting if something appears. But yeah, in terms of science, it would be possible, but I think it would take way too long. We need to find another way. Okay. And also, I also think that if you do it in a laboratory, the experiment would be small. I mean, you need a very big environment to make this happen. You cannot just take a little bottle or a volumetric flask and put all the necessary things in there and then expect that life appears after a while. Because, I mean, we're talking seas on planet Earth, and we're talking really, really big environments where a lot of things can happen, and we don't know exactly what happened at which moment. So to do this exactly the same in the laboratory, it might be difficult. So let's go to the bacteria and to the other things that we're going to talk, the bacteria and the cells that we're going to talk today about. I want to tell you that in the feedback, you told me that you would like to have some more specific information. So I'm going to share this presentation with you. I'm going to upload it so you can consult it if you want to have a little bit more detailed information. So let's have a look at the cell. Guys, it's normal that you will not understand everything, but that's not the idea of today. The objective of today is just that you recognize the cell parts and that you know more or less what a function is, okay? We will spend the whole second partial to see each cell organelle. Organelle is a cell part. We will discuss that in way more depth. So this is just an introduction. It's just to show you which parts we have and that you can map it in a cell, that you have an idea what we're talking about, okay? So don't get scared about this. It's a very theoretical class, but the objective is just you recognizing certain things. I also know that many of you already got some introduction about this in secondary school, so I don't think it's too hard. But again, it's not necessary that you understand every little detail of every little cell part. That comes later. We just want to recognize them and know their major function. That's the objective of today, okay? And when we do that, there is two things, two types of cells that we are going to have a look at, okay? We have the prokaryotic cell and the eukaryotic cell. They're not on scale. So you can clearly see here, if you look to the micrometers, the eukaryotic cell is more than 10 times bigger than a prokaryotic cell, okay? Even 100 times if you have a very small one, or even 1,000 times could be if you have 0.1 to 100. So prokaryotic cells are simple cells, okay? This is the cells that you find in bacteria, archaea, which are algae, and other single cell microorganisms, okay? The eukaryotic cell is more complex cells. This cell came way, way later after the prokaryotic cell, and we believe that the eukaryotic cell was formed out of combining several, one or two or even more prokaryotic cells. And the eukaryotic cell is what you see on my background. This is plant cells and animal cells. And we will mainly focus on the eukaryotic cell. And why is that? Because the eukaryotic cell is a big and a complex cell. And everything that you find in a eukaryotic cell, you will also find it in a prokaryotic cell. But what you find in a prokaryotic cell, you don't find it in a eukaryotic cell, okay? Because it's way more simple, okay? So by understanding the eukaryotic cell, you will have no problems understanding the prokaryotic cell because it's way simpler. What do they have in common? That's our first slide, okay? All cells have cytoplasm, okay? Cytoplasm is a liquid inside each cell, okay? And inside this liquid is necessary because it contains nutrients, it contains biomolecules, it contains everything a cell needs. I don't like to compare it like this exactly, but you could say a little bit, it's of course not the same, but you could say it's the blood of the cell, you know? If there is nothing inside the cell, there cannot be air in it either, you know? So that's why you can see it a little bit as the life blood of a cell, you know? It's where everything is floating in it, in the cytoplasm. Then, secondly, all cells have a plasma membrane. We already told in previous classes that this plasma membrane is made of a special biomolecule, which we call phospholipids. We will see that more in detail, how that exactly works later, but you just have to know that each cell has a fatty membrane. It's a phospholipid membrane, okay? That's also why soap is so efficient in killing bacteria, okay? If you have bacteria on your hands, Bacteria have a plasma membrane, and if you add soap to fatty things, you all know that the fatty substance disintegrate. The same happens to bacteria. By using soap, you destroy basically the plasma membrane, which is like a little fatty layer around the bacteria, and they die. Okay, that's why we have soap, and that's why it cleans us. Okay, um, so all cells have this membrane. Okay, plasma membrane. All cells also have DNA. Okay, all cells have DNA. Not by no certain viruses. Not there's some exceptions, but you just remember that all cells have DNA. Okay, DNA is basically the biomolecule which we call. Who remembers what's the name of the biomolecule DNA? In which group does it go?
Nucleic
Amino
acids.
acids.
Exactly, nucleic acids. And they also have RNA. Okay, the difference between DNA and RNA is that DNA is double-stranded and it's safe in the nucleus. In bacteria, it's floating around a little bit. And the RNA is shorter and single-stranded. You can compare it with DNA being the library of all knowledge and all blueprints of the cell that is in the nucleus, if we talk about eukaryotic cells, and the RNA is the copies you make of that book. So it's a copy, a photocopy. It's just 1 or 2 pages that you copy the parts that you need of the knowledge of the DNA of the cell. Okay, is that clear? Okay. Guys, now forget a little bit about this prokaryotic cell. We're going to look at the eukaryotic cell, okay? So we're not talking about bacteria anymore. We go to plant and animal cells, and I'm going to show you 2 pictures of it. This is an animal cell, and this is a plant cell, okay? Have a look at it, a plant cell and an animal cell. And now my question is, what are the main differences? There are not many between plant and animal cells, but you can immediately see certain differences. Who can mention me one?
There's a lot of.
So
Sorry?
their cell wall is different.
Yes, very
The
good.
cell wall.
No, it's not different, Ana Sofia. It's not different. But you're right. It's not different because an animal cell has no cell wall. All cells have a plasma membrane, okay, or a cell membrane, but they don't have a cell wall. So an animal cell has a cell membrane, what you see here in beige brown, and a plant cell has a plasma membrane as well around, and it also has a cell wall. So animal cells do not have cell walls. What is the difference between a cell wall and a cell membrane? A cell membrane is made of phospholipids, and a cell wall is made of another biomolecule of the implants, usually the carbohydrates group being cellulose. Okay, so think about a piece of wood. Wood basically is cell walls. Okay, wood is basically cellulose. And if a plant would not have cell walls, then a tree could never be as strong and as tall as they are. A plant would not be able to grow. It would just be, they will fall apart, you know? You understand this?
Yeah.
We humans are different, you know. Our cells are connected with proteins and everything. But imagine you take a piece of meat. You cannot stand a piece of meat straight, you know. If you take a piece of meat, if you do the carnita asada, it would also fall down, you know. It would fall down and would like a little ball, you know. So it doesn't have the same strength, and that's because of the cell walls. Because meat doesn't have cell walls. Okay, plants have. And that's why a piece of wood is strong, or you take a little branch of something. That's because of the cellulose. So cell walls are made of carbohydrates, usually cellulose. Okay. So second difference, Luna, what do you think? If we have a plant cell and animal cell, you can immediately see one big difference here.
The tiny purple holes.
Tiny purple holes. You mean these ones? Here, this one?
Yep.
They have it both, but yeah, I do agree that here it's not so obvious. But no, they both have it, in fact. But here it's not.
The central
here is a
vacuole.
Yes, very
Big
good.
thing.
Central vacuole is a big difference between animal cells and plant cells. Why animal cells do not have vacuoles? It's very simple, because we have to eat regularly, and that's how we get our nutrients. Plants and trees work slightly different. They have to suck up the nutrients through the water that they absorb with their roots. And of course, those nutrients have to be stored somewhere, okay? Because depending on the plant, it's not always readily available. Moreover, plants are usually bigger, and trees as well. They can be way, way bigger than humans. So these nutrients have to travel a longer distance. So they have to be stored somewhere. And that happens in the vacuole, okay?
Plants also have chloroplasts, which kind of goes hand in hand.
Yes.
So they do photosynthesis, they produce their own food, while
Yeah, go ahead, Anna Sophia. It's
they don't
perfect.
have the photosynthesis.
Yeah, okay. We can go
Yeah,
there.
the animals using sunlight as an energy source.
Perfect. You know a lot about it, indeed. Let's stay for a moment with the central vacuole. So what do you think, guys, if you have a desert plant and a jungle plant, which plant has the biggest vacuole?
Jungle.
Desert.
Yeah, the desert plant, of course, because there's less rain available, there is less liquid available, so the plant has to store more for the times when there is no water or nutrients available. So typically jungle plants, they have smaller vacuoles. Okay, then Anna Sophia said another interesting thing, and that's this green thing here, the chloroplast. It's really one of the most incredible things of evolution. The chloroplast, it's undeniably a magical thing, because it's thanks to the chloroplast that we are alive. Without plants, we wouldn't be here. Okay. Because we complement each other. What's so special about those chloroplasts is they are, like Anna Sophia already said, they're able to capture sunlight, and sunlight is the source of energy, and they indeed do photosynthesis. And as the word says, photo refers to the sunlight, synthesis means to produce something. So what a chloroplast is doing, it's taking CO2 out of the air, and with the energy of the sun, it can convert different kinds of different amounts of CO2 into a carbohydrate, usually glucose. Okay, and this is a process which we call photosynthesis. Okay, and this glucose can be converted in in anything you want. It can be converted in cellulose, it can be converted in strawberries, in leaves, in fruits, in whatever you want. So that's a really, really amazing thing what plants do. That's why it's thanks to plants, it's thanks to the process of photosynthesis, and it's thanks to the chloroplasts that that the atmosphere of the Earth is what it is today. Okay, it's in fact life that took CO2 out of the atmosphere and produced oxygen as a byproduct. Okay, without that, we wouldn't be here, because we breathe oxygen, and then the atmosphere would still be maybe something more like Venus. So it's thanks to life itself, thanks to plants and algae and photosynthesis, that life became possible for us. It's something really mind-boggling to think about this. And that's why I'm very fascinated by this, because in fact, think about it. Trees produce oxygen and we breathe oxygen, okay? Trees produce glucose, okay? And we consume glucose. I mean, we're completely opposites from each other. Plant life and animal life, it's complementary. We cannot, I mean, the plants can live without us, but we cannot live without the plants. If trees wouldn't be there anymore, we will be done very, very soon. That's why we should plant as much trees as we can and save the Amazon forest. I can get very emotional about this, because it's something that I don't understand why we allow the world to be so destructed if this is the essence of life. Without trees, we're done. Really, we're completely done. I hear people in Saltillo, I know people in Saltillo No, es que no me quité los árboles en la banqueta porque se sube la banqueta. Please, how can you remove trees porque se sube la banqueta? I mean, it's impossible to think this. We need trees. If they bring insects or whatever, it's not an excuse. Without trees, we are completely done. And you see that we already get chronic illnesses just because in our cities there is just not enough green anymore. But anyway, that's an whole other topic. Um, let's go to the cell components. So I'm going to skip here a bit. You don't have to know this all. I just want to go to the cell components. And let's go to the first most interesting one of every eukaryotic cell, which is the nucleus. Okay, the nucleus is easily observable. You can see that under the virtual microscope. We've seen that. It's like a dark dot. And the nucleus has several parts, three big parts to be correct: the nuclear envelope, the chromatin, and the nucleolus. Okay, let's have a look into each one of them. The nuclear envelope is basically what surrounds the nucleus. Okay, it's just like a membrane covering the nucleus. The nucleus itself has chromatin. Okay, chromatin is basically DNA. It's DNA with proteins. Okay, think about your grandmother who is sewing. DNA has to be packed because it's really, really long, and if we don't pack it correctly, it would be a complete mess. So chromatin is DNA and histones together. What do the histones do? We just use them for the DNA to be wound upon. Okay, so that the DNA is nicely packed. I have an image of that here if you want to see that. So here you see DNA and here you see the histones. Okay, what happens is that the DNA is all wound around those proteins to pack it nicely inside the chromosome, you know, that it forms a chromosome. Because otherwise, think about it, if you have a thread of a very long length, yeah, and if you don't pack it nicely, it would be a complete mess full of knots, and you cannot have that with DNA. It has to be nicely organized, and that's why we have these kind of histones that help to pack the DNA as to keep it nicely organized. Okay, so chromatin is basically DNA together with the proteins in order to save it nicely. Okay, then we have the nucleolus, which is like the center of the nucleus. This nucleolus doesn't contain DNA. It is the place where ribosomes are produced. Okay, ribosomes are produced in the nucleolus. Okay, it's made out of ribosomes are made out of RNA, some proteins, and they're produced here. And whenever they are produced, they travel through the nucleus to the nuclear pore, and then from the nuclear pore, they go to the endoplasmatic reticulum, and there they stay on the membrane. Some of them, they float freely into the cytoplasm, but the ribosomes, they usually stay, the big majority, on the endoplasmatic reticulum because their function is to produce proteins. Okay, so but we go there in a moment. Is the nucleus clear for everybody? Yeah?
It's made of
Yes, yes, yeah.
It's made of what? Sorry.
It's made, what is made of what?
The third one. Sorry.
Sorry, I didn't catch that. Can you repeat yourself, please?
I think the nucleolus.
The nucleolus itself. It's like a part of the nucleus, and it's a membrane, let's say. It's like a core inside the nucleus where the ribosomes are produced. And the ribosomes are, in fact, little small combination protein with RNA, and they produce proteins. Okay, but don't go too much in detail because you make it unnecessarily complex. But we see in a second what they exactly do. You just have to know that those ribosomes are produced inside the nucleolus, and the nucleolus is in the middle, in the center, inside the nucleus. Okay.
And what's the function of the nuclear envelope?
The nuclear envelope is a membrane, which you can see here, is the membrane that keeps the DNA together. So it's basically, you can compare it with the cell membrane of the nucleus. If there would not be a nuclear membrane or a nuclear envelope, then the DNA would spread out all over the cell. It's like the entity that keeps it together. Okay. And then the nuclear pores are little holes into the nuclear envelope, and those little holes are necessary because the ribosomes have to leave the nucleus, otherwise they cannot leave. Okay. And also things have to go inside the nucleus to make copy of the DNA. So it's like little doors, if you want, to go inside and outside the nucleus. And of course, they're guarded doors because not everybody is allowed inside the nucleus. Okay, it's like the most important part of the cell. Okay, let's go to the second part. Here you see a ribosome is a small particle composed of ribosomal RNA and proteins. Okay, and what those ribosomes do is they produce. They are made in the nucleolus, and they produce. It's like the machine that makes the protein. Let's have a look how that works. Okay, but first let me explain where they're producing. Okay, so what is next is here you have the nucleus, and the nucleolus is in the middle, and then we have what we call the endomembrane system, and the endomembrane system has 5 things: vesicles, the endoplasmatic reticulum, the Golgi apparatus, the lysosomes, and the vacuoles. Okay, if we look to each one of them, you will have a better understanding why they call it endomembrane system, because they are all made of pieces of membrane, and each of one has a specific function. Vesicles, they make sure for transportation. Okay, endoplasmatic reticulum is the place where proteins and phospholipids are produced. Don't worry yet, we'll go there in a moment. The Golgi apparatus is the place where the proteins are packaged and assembled for transportation. The lysosomes is like a little vesicle with digestive enzymes in it in order to recycle things that the cell doesn't need anymore. And vacuoles is what we have seen already in the plant cell, is to store things. Okay, so vesicles is like a little bubble inside the cell. Okay, and when the bubble goes from inside the cell to outside the cell to remove something from the cell, we call that exocytosis. When the bubble takes something from the environment outside the cell inside the cell, so like ingestion, we call that endocytosis. Okay, so endo in, exo out. Okay, that's what vesicles do: transportation inside the cell, but also with the environment outside the cell. So if something has to be removed from the cell, a vesicle fuses with the membrane and releases it in the extracellular matrix, and it's gone. Okay, if the cell needs some nutrients or something has to be absorbed, then endocytosis kicks in, and the vesicle goes from outside inside the cell. In fact, it's a piece of membrane that eats like Pac-Man, and then it blobs off and it goes inside the cell. Okay, endo and exocytosis. Then we have the endoplasmatic reticulum. Okay, the endoplasmatic reticulum is directly connected to the nucleus very often. Okay, usually it's always directly connected, like you can see here. Okay. We have to make a small distinction here because we have smooth endoplasmatic reticulum, you can see here, which is more on the edges. And then right next to the nucleus, we have the rough endoplasmatic reticulum. Okay. Why is there a difference? It's because, like you can see on this picture, the rough endoplasmatic reticulum has like these little balls on it, these little dots. Those little dots are the ribosomes who come from the nucleolus. Okay, and there is a part of the endoplasmatic reticulum who has not these little dots on it, and that's what we call smooth. They call it smooth because under the microscope, the smooth one, it looked like a smooth membrane, like you can see here, and the rough one, it would have those little dots, so it seemed a rough or a not smooth surface, and that's why they gave it the name smooth and rough endoplasmatic reticulum. Okay. It's also important to make the distinction because the rough endoplasmatic reticulum, which is the red one here, okay, this one has ribosomes on it, and this one is producing proteins, okay? While the smooth one, it has no ribosomes on it because ribosomes are involved in the synthesis of proteins. So the smooth one is involved in the production of lipids. For instance, the phospholipids, where the cell membrane is made of, are produced in the smooth endoplasmatic reticulum. So if a cell wants to grow, it needs more phospholipids. Those phospholipids, in order for the membrane to grow, they come from the smooth endoplasmatic reticulum. Okay? Everything clear so far? Okay, then we have another part which is not directly connected to the nucleus or to the other parts. It's like a small different other entity. It could be connected with the endoplasmatic reticulum in certain cases. And this is what we call the Golgi body or the Golgi apparatus. So whenever the endoplasmatic reticulum, the rough one, the rough endoplasmatic reticulum, produces proteins, those proteins are not 100% ready yet. They still need to be packaged, they still need to be assembled, they still need to be folded. There has to be certain final touches to be done with. And this happens in the Golgi apparatus. And you can see it a little bit like the post office. Yeah. What happens is they receive all the products, they give it the final touches, they package it. The packaging is the formation of vesicles. Okay, this is the Golgi apparatus producing the vesicles and send them away for transport. So it's not the Golgi apparatus who is doing the transportation. That's what the vesicles is doing. The Golgi apparatus receives every product that is synthesized, does the final touches, and prepares it for packaging and sends it away to the right place where it has to be. Okay, very simple. It's like the post office of a cell. Okay, and then the last thing is what we have is the lysosomes. Vacuoles, yeah, that we already talked about. It's storing material and nutrients. And then we have the lysosomes and the mitochondria. So the mitochondria is the power plant of a cell. That's the only thing you have to remember for now. It's what produces the energy of a cell. We're going to see that in much more detail later. And the lysosomes is the part of the cell where the recycling plant, okay, where things that are have to be digested or the cell doesn't need anymore are recycled and sent away for destruction. So here, to finish for today, I quickly want to do with you this activity because this is the very essence. If you do this activity, then what we learned today, that's all you have to know. More details, not yet. We go there later, okay?
But I have a question really quick. Like, what's a quick explanation of the vacuoles?
Vacuoles, it's a storage unit for the cell, for plant cells, specified for plant cells. It stores waste products, it stores nutrients, it stores water, whatever needs to be stored. Okay, so here we have function cards. We have memory items. I have the answer key below. I will share that with you. Basically, what you have to say, what is the function of the cell membrane? And we're going to take one here and we're going to put it right there. Okay, very simple. So what captures energy from sunlight and produces it to, and it uses it to produce food in plant cells? Luna? Oh, yeah, Ana Sofia, fine. Sorry.
Yeah, the chloroplast, sorry.
No, it's fine. I'm glad you participate. Chloroplasts, very good. This is chloroplast. Okay, so this one goes here. I'm going to share this with you. You don't have to do it. It's just like we remember it. Controls what comes into and out of a cell found in plant cells and animal cells, both. So don't tell me cell wall, because that's only plant cells.
Membrane.
Yes, Ana Pao? Sorry?
No, it's okay. It's okay. No.
No, but you know the answer. Come on.
Golgi apparatus?
No, it controls what comes in and out. So if you think about it,
Endoplasmatic.
Cell membrane.
Membrane.
yeah, the cell membrane, of course, because it's the first border of the cell, you know. So that's what surrounds the cell, and that's what's telling what comes in and out of the cell. So you can see it here behind me. In the plant cell is like a membrane. The cell membrane is made of phospholipids, and that's what controlling what goes in and what goes out. What is the shell-like fluid where the organelles are found? So we saw that in the beginning of the class.
Cytoplasm.
Cytoplasm.
Cytoplasm, very good. What is the control center of the cell which contains DNA? Very easy one.
Nucleus.
Nucleus, don't confuse with nucleolus, okay? Nucleolus is where the ribosomes are produced, and nucleolus is a part of the cell, of the nucleus. So found outside, inside the nucleus and produces ribosomes, I just said it.
Nucleolus.
Nucleolus, okay, perfect. How do we call the rich outer layer of a plant cell?
Cell wall.
Okay, cell wall, very good. Made of the biomolecule carbohydrates of that group, more specifically, usually cellulose. What uses chemicals to break down food and worn out plant cell parts? Sorry, not plants, cell parts. So the recycling plant of a cell.
Lysosome.
The lysosome, yeah, really good. Then what receives proteins and materials from the endoplasmatic reticulum, packages them and distributes them?
Golgi
Smooth.
apparatus.
The smooth.
Yeah, it's the Golgi body or the Golgi apparatus, yes. Okay, produces the energy a cell needs to carry out its functions.
Mitochondria.
Yeah, perfect. It's the power plant of the cell, okay? What is assembling amino acids, because proteins are made of amino acids? What assembles amino acids to produce proteins?
The rough ER.
Yeah, correct. Yeah, it's the rough endoplasmatic reticulum would be fine, but more specifically, indeed, the rough endoplasmatic reticulum. What does store food, water, waste, and other material?
The vacuole.
Vacuole, okay, we're doing great. Has passageways that carry proteins and other materials from one part of a cell to another?
Vesicles.
Yes, vesicles. And then the last one, tiny strands inside the nucleus that contain the instructions for directing the cell's functions.
Ribosomes.
Would that be DNA? Or
Yeah, that would be DNA indeed.
Chromatin?
DNA or yeah, that's indeed the genetic code. So here you have it. It's a chromatin. Okay, DNA would be fine, but chromatin is also fine because chromatin is DNA with this. So I'm going to share this for you. It's like a very handy tool to remember all those parts. You know, I'm the powerhouse. I clean things up. I'll store anything. I'm a golden packer. So it's like a very ludic way of learning this. So I share with you this. I shared this with you on Canvas, and this is everything I planned for today. In fact, so I wish you quite a nice afternoon, and we see each other tomorrow. Do not forget, we see each other at 11, okay? And it will be our guest speaker that will be talking to you, not me. Maybe I give a brief introduction, I don't know yet. But you connect to the same Zoom channel, and hopefully you enjoy our guest speaker, okay? The idea of the guest speaker is to be inspired and to be convinced that you can do anything if you just want it enough, okay? So take care, guys. If you have any questions, you can still ask. Otherwise, you're free to go.
Thank you.
Thank you.
Thank you.
Okay.
Bye.
Thank you.
Bye.
Thank
Take care.
you.
See you.